Hey guys, Daniel King here with Dr. Blair's BY403 Immunology course. Today I'm going to be discussing three different terms. Those are isotypes, allotypes, and idiotypes. The first thing I'll discuss is isotypes. Those are basically an antibody class that is determined by basically the constant region sequence of the heavy chain. So antibodies consist of a light chain and a heavy chain in their structures. In the case of isotypes, humans in particular have 5 different isotypes. Those are defined as IgA, IgG, IgE, IgF, and IgM. These isotypes have both differences in their structure and functions. So the heavy chain on these particular antibodies are different in structural conformation, and they provide different functions within the immune system. In particular, isotypes have been shown to be very effective in antigens when used for immunizations. This is more important in the case of infants. Newborn infants have a relatively underdeveloped immune system, and introducing isotypes into their immune system can help them develop and proliferate their immune system to be more advanced and adaptive to different antigens and pathogens within the environment. The next term I will be discussing is allotype. Allotype is basically alternative forms of isotypes, but they're still the same isotype. So it's just differences in the immunoglobulin chains of each isotype, so they're basically the same. A good example of where allotypes are important are between animals of the same species. So one animal can have the same isotype as another animal, but they may be considered allotypes. Or in the case of transferring immunoglobulins from one animal of the same species to another animal of the same species, that can be beneficial in the aspect of just further developing and toning and honing the immune system of the different animals of the same species. So introducing allotypes within the immune system from one animal to another can benefit in basically building more antibodies against antigens from particular pathogens. Finally, the last term I will discuss is the idiotype. It is a set of antigenic determinants that can characterize unique antibodies or T cell receptors. These are more of a newer discovery, and they are currently used in a lot of clinical labs and tests against newer diseases or previously uncurable diseases. An example being something like lymphoma. Scientists have been trying to create vaccines against diseases like lymphoma, and they are successful when using the idiotypes of antibodies. And perhaps within a couple of years or a few years, we can further develop this research, and it will be able to be, the disease will be able to be treated with vaccines. So that's it for those three terms. Thank you for listening, and I hope you learned something about isotypes, allotypes, and idiotypes.